I'm Emme.
And I'm Vanessa.
Today we're going to teach you a little bit about the cell cycle. First, we're going to teach you about interphase. Interphase is the longest part of the cell cycles where the cell practically doubles everything in the cytoplasm. In the first part of interphase, the cell grows and prepares. It increases in size and makes proteins and molecules necessary for the cell to function, and some of the organelles begin to duplicate. The second part of interphase is called replication. The DNA, which is inside the nucleus, contains sides of sugar and phosphate, and there are 4 steps made of the 4 nitrogen bases called thymine, adenine, guanine, and cytosine. During replication, the DNA molecule is unzipped or unwound by an enzyme. The sides of the latter separate, and each side becomes a blueprint for the 2 identical copies of DNA. Free bases floating around come and pair up along the 2 unwound DNA sides. A pairs with T and C pairs with G. After this is done, there are 2 identical copies of DNA. This is important because when the cell splits, they will each need a copy of DNA, and the cells need to be identical. In the last stage of interphase, the cell continues to grow and prepare. It grows and continues to make proteins that will be used for the new cells formed in cytokinesis. In this stage, the chromatin is loosely coiled, so sections of it can unwind for RNA to be made. The chromatin contains the replicated DNA that is copied into RNA. The RNA is used to make proteins. During continued growth and preparation, organelles such as the mitochondria and chloroplasts are duplicated in anticipation of the new cell. The next part of the cell cycle is mitosis, which is normally the shortest stage. In the mitosis stage, the contents of the nucleus divides. As the nucleus prepares to divide, the DNA molecules that are replicated during interphase join together into sister chromatids of chromosome. They are joined together by the centromere. There are 5 parts to mitosis: early prophase, late prophase, metaphase, anaphase, and telophase. The first part of mitosis is early prophase. In early prophase, replicated chromosomes coil into X shapes and become visible under light microscopes. The nucleosis disappears and the nuclear membrane begins to break down. Spindle fibers, or tiny tube-like structures of protein, begin to form. The spindle fibers stretch across the cell from centrioles, which are organelles that organize spindle fibers. In animal cells, the centrioles have migrated to the poles of the cell. Stage late prophase, the nucleus has disappeared and the nuclear membrane is gone. Spindle fibers are fully formed and chromosomes attached to spindle fibers at their centromeres, then the nuclear membrane disappears. Third phase, metaphase, a tugging force of the spindle fibers pulls chromosomes into a line at the cell equator. Stage anaphase, spindle fibers begin to contract and shorten. The centromere pulls apart and the sister chromatid are pulled apart to opposite ends or poles of the cell. After being pulled apart, each sister chromatid is now considered a chromosome. Final stage of mitosis, known as telophase, one complete set of chromosomes is at each pole of the cell and the spindle fibers begin to disappear. A nuclear membrane forms around each chromosome set and the nucleolus appears. The result of all this is that there are 2 nuclei in one cell, so it is ready to divide. The final part of the cell cycle, called cytokinesis, the 2 nuclei are split into 2 daughter cells that are identical. In animal cells, the cell membrane pinches together to divide the cell's cytoplasm and organelles. In plant cells, a cell plate forms along the cell's center and divides the cell.
Thanks for watching. Make sure to subscribe to McCrae Science 2016.